today's lecture. Good morning, ladies and gentlemen. I'm very glad to be here to attend this international academic exchange. The topic I am about to share today is molecular markers in vegetable breeding. And I am William. And some of you may still remember me because we have met 1 year ago. Right now I can recognize 4 of you that have attended this lecture last year. 1, 2, 3, 4. Yes, 4. 2 gentlemen and 2 ladies. And since this becomes a review for you, I'm going to ask you to be a group leader on the experimental lesson that we are going to have this afternoon. So be prepared. You are going to be group leader this afternoon. And for those who are first time to attend this lecture, I am pretty sure that I will give you a better lesson than the one last year. First of all, as a guest from China to your country, I'd like to give a brief self-introduction to you. This is my Chinese name, composed of 2 Chinese characters which resemble each other, but they are pronounced differently, Li, Wei. And that's why I choose William as my English name. And I was born on April the 1st, 1988. As you know, April the 1st is a Fool's Day, the day that people play tricks. So it was very hard for me to receive a happy birthday gift from friends, because when I told them it's my birthday on April the 1st, they say, haha, funny. And from 2009 to 2015, I studied in University of Science and Technology of China to get my doctor's degree. After that, I worked as a postdoctor in Anhui Jianghuai Horticulture Company, which is the organizer of this workshop. Now I am a teacher of a School of Horticulture of Anhui Agricultural University. The research work I have been engaged in the past 5 years mainly focus on application of molecular markers in vegetable breeding. Vegetables are vital for all human beings on this planet. Maybe there are some people who are vegetarians. They can keep away from meat, but no one can live without vegetables, which can provide vitamins that our body cannot produce for ourselves. And vitamins keep you from getting scurvy, which is a severe disease that may kill you. So we invested much manpower and material resources into vegetable producing. Chinese vegetable production value has reached over 500 billion yuan 3 years ago. This is a giant market. To meet the vigorous growth of vegetable demand, new varieties and better techniques are in urgent need. So we also invested much manpower and material resources into vegetable breeding. We want to get new and better varieties. Actually, in China, the production, supply, marketing of vegetables is a business with the largest number of employees of all businesses nationwide. This leads to over 30,000 vegetable germ plasmas reserved in National Clonal Germ Plasm Repository. With so many labors invested into this territory, guess how many works have been done by them? Only 3 things, actually. Selection, hybridization, and production. These are done respectively by breeders, selection, and producers of hybrid seeds, hybridization, and producers of vegetables, production, to bring a consumer-favored variety of vegetable into market. First, we need vegetable breeders to select, the first step, selection, to select a desirable maternal inbred line which has high productivity, for instance, and then select a good paternal inbred line which has great disease resistance, for instance, and producers of hybrid, the second step, hybridization. Producers of hybrid seeds hybrid this good maternal line and this good paternal line to produce F1 seeds. F1 generation not only inherits all those good characters from both paternal and maternal lines, but also surpass parental materials. This is a phenomenon called heterosis, hybrid vigor. One of the most critical matters producers need to pay attention to is that they must ensure all the seeds that they sell are true F1 seeds with hybrid vigor. In other words, purity of F1 seeds must be ascertained to meet the national standards before the seeds were sold to farmers, which are the producer of vegetables. The third step, which mainly involves cultivation techniques, is not included in today's topic. I believe other teachers may have talked on that topic before. Today we focus on the first 2 steps, showing you how do farmers and breeders do in the past and how do we do nowadays with molecular technology. In the past decades, like most other countries' vegetable breeders, we accomplished the first 2 steps, selection and hybridization, mainly rely on visual inspection and experiences. Let's run through the first 2 steps. First one, selection. Let's say your goal is to select a pepper variety with increased productivity. It usually happens like this. One day, you find a bigger one among ordinary ones in your field. You have to collect seeds from this bigger pepper, and then you plant all the seeds of it and nurture all the seedlings until fruit setting. That is when you can tell which pepper is bigger and which is smaller. Because not all the progenies manifest the exact characters as their ancestor due to the heterozygosity of this bigger one's genome. it is not pure. It has heterozygosity in its genome. So the progenies will differ between each other. Then you can select the biggest one, this one, this one, this one, from F1 generation, and repeat the process. You collect seeds from those bigger ones and you plant them again. You repeat this process for several generations, and then you get yourself a big pepper inbred line. After several generations, the phenotype becomes stable. This is actually a classical breeding method adopted by breeders all over the world for decades. But the greatest disadvantage of this traditional breeding way is labor and time consuming. You can imagine that, right? You have to plant all the seedlings, all these seedlings, bigger one and the smaller one, all these seedlings into field and nurture them until fruit setting. That is when you can tell which is bigger and which is small. One repeat of this process will cost you at least half a year. And usually it will take 6 times of repetition of this process to get an inbred line. Besides, it's critical to provide uniform growth environment to all these F1 generations so that the genotype of every single seedling can display stably. Otherwise, it will be influenced by the environment. And a plant with high productivity potential would encounter production reduced if it was planted in a sterile area of the field. So the environment should be uniform. This would cost you many money. This is the first step, selection. It is labor consuming and unstable in traditional way. The second step, hybridization. After selecting desirable parental materials from the first step, now you can hybridize. Hybridize the good maternal inbred line and the paternal inbred line to get F1 seeds. These seeds will grow into plants with advantages of both parental materials. But during manufacturing of F1 seeds, failure would sometimes happen due to predehiscence of the maternal material stamen and inactive stamen of the paternal material, which brings seed purity issue to manufacturers of F1 seeds. And this brings the problem of seed purity. The seed law of People's Republic of China ruled that the seed companies will face very heavy punishment if they sell seeds with purity lower than 95%, which is a very high level. So seed purity must be checked cautiously for every batch of seeds. Even though cost of seed purity identification is very high. Like the first step, you have to plant them into the field and culture them until fruit settles. So the seed purity identification is time and money cost, and the labor and time consuming work. And even though the results would be usually unstable due to the change the environment. How can we overcome obstacles in these two steps? The answer would be molecular markers. When you are selecting desirable plants, you think you are selecting phenotype, but actually you are selecting genotype during these two steps. Because everything about plants and other animals, even including people, is controlled by genes. Since genes won't change as seedlings grow into mature plants, we can conduct detection of genes at seedling stage to inspect which ones of them inherit good quality traits from parental materials. Then we can give meticulous care to them and get rid of the others, which can save you a bunch of time and money. But why do we use markers, molecular markers, instead of directly using genes in vegetable breeding? Because we just said that all the phenotypes of vegetables are caused by gene. Why don't we directly use gene in vegetable breeding? Instead, we use molecular markers. What are markers? Markers are something that can assist us to ascertain the genotype of the plant. Genetic markers are repeat DNA sequences distributed uniformly within genome, and the locations of genetic markers on a chromosome is known. Two important things. DNA markers are distributed uniformly, the first important, and the second important is their location is known, which can be used to locate genes. However, gene sequences differ greatly with each other, and location of genes are unknown, unlike markers. It is an extremely difficult task to locate a gene. I'll give you an instance to illustrate this. Let's take a look at this map. This here is my university, Anhui Agriculture University, which resembles a gene. And you want to get to the gene. And you asked me how to get there from here. That is the process that you locate a gene, right? And it would be very difficult to describe a location in the city because all those buildings, they look similar with each other. You can't tell which building is the university. So it's difficult to locate the building. But there are some markers which distribute one uniformly within the city. The markers are distributed uniformly in the city that we can use to locate the university. And the second is location is known, resembles two characters of markers. What is this marker in the city? The subway station, as you all know. So the feasible way would be telling you to take subway and get down at the 7th station. Genetic markers are just like stations, which distribute uniformly in the city of genome, and their locations are known. Using markers to assist selection is called MAS, marker-assisted selection. Let's take a look at the advantages of MAS. First, it is more reliable than inspection ways. One major disadvantage of subjective measure is that feelings of people are not the same. Even sunlight can easily affect your evaluation on skin color of a fruit, right? On the contrary, math is much more reliable because sunlight won't change DNA. And second, math can overcome the influence problem of the environment. Even in the same field, some area is worse than other area. But no matter how environment changes, markers won't change. And third, you can conduct early selection during seedling stage. Then you can know which seedling is the one that you want, and you get rid of all the others, which can save you much time and money. Fourth, improve selection intensity. When the selection becomes very quick with mass, you can increase selection population to a very, very large population to get more desirable plants within just one generation. Last one, fifth, non-destructive examination and selection. For example, when selecting anti-rice blast phenotype, you have to inoculate bacteria into the rice, into the plant to accomplish examination. When you finish the selection and get plant strains with good anti-stress phenotypes, they are already being damaged by pathogens to some extent, right? But with DNA mass, you don't need to inoculate bacteria into plants. So it's non-destructive examination and selection. Okay.
In number 5, for example, if I am selecting seeds, well, I suppose that I have to some way to extract DNA from seeds. So, are non-destructive ways to selection in seeds, or, for example, to accomplish desired purity in F1 generations, or no, you have to take seeds and extract your DNA?
Yes, very good question. You mean that the DNA extraction is actually some destructive to some extent to the F1 generation, right? But you don't have to extract DNA from seeds. You can plant them and extract DNA from seedlings. You just take one leaf from it, so it's non-destructive. Compared to the traditional way, you have to inoculate bacteria into the plant. The whole plant is severely damaged by bacteria, even though you can collect seeds from it, but the plant has already been severely damaged.
Thank you.
You're welcome. Yeah, that's a very good question, and for the others, if you have any questions, you can interrupt me at any time, okay? You don't have to wait for me to this again. Let me give you more details about these 5 advantages, since you are interested at this part. Could you please tell me which one is greener? If you actually, the color of the skin of the fruit is actually a very important character, because consumers, they pick up peppers in the supermarket, they tend to choose those ones that looks healthier, right? It's greener. That would be a very good character. But if you want to select an inbred line with greener skin, so the traditional way would be to evaluate the skin color by inspection. And maybe someone would say that this is greener, maybe someone would say this is greener. It depends on the environment that you do the inspection, right? And second, you all know that in the field, there are some areas in the 4 corners. In the 4 corners is the area that can get dry easier than the central part, right? At the 4 corners, plants won't grow as better as those plants in the central. So it will be very easy for you to evaluate the F1 generation, saying that these seedlings have great vigor than those corner ones. But actually they are not. They're just influenced by the environment. Third, you can employ early selection during seedling stage. You don't have to take the whole leaf. Actually, all you need is just like this small portion of the leaf is enough. Now, since mass has overwhelming superiorities over traditional selection, scientists have developed many kinds of markers for mass. Yes, mass is not a single method. Based on different markers, several kinds of mass are developed one after another. Frequently used mass include many kinds, like RFLP, ISSR, ASLP, SMP, and of course, the main character of our lecture, SSR. We need to choose a kind of marker considering large-scale application needs. Detection of the marker must be easy. We chose SSR. It can be detected very conveniently. SSR represents simple sequence repeat. It is a tract of repetitive DNA in which certain DNA motifs ranging in length from 2 to 5 base pairs are repeated for typically 5 to 50 times. This is a GA repeat example. You can see the sequence of this DNA is GA, repeated for typically 5 to 50 times. There are also other kinds of repeats. For instance, AT, GA, TC repeats, and repeats of 3 characters, AC, C, CTC, and repeats of 4 or 5 nucleotides, GATA, TG, CA, like this. 2 characters of SSR are that SSR occurs at thousands of locations in the genome. So there are a bunch of them. And they are notable for their high mutation rate and high diversity in the population. This is very important. The marker has to be with high mutation rate and high diversity. Otherwise, the SSR marker, if it's the same between paternal and maternal, it will be impossible for you to use the marker to identify which is maternal and which is paternal. So this is very important. It has high diversity in the population. The underlying mechanisms of mass with SSR is that different genomes have different SSR lengths. So, like this is SSR from paternal and this is FFR from maternal. After PCR and electrophoresis, you get this. Then positions could reflect polymorphisms among plants. So with only one step of PCR and one step of electrophoresis, it's so easy to detect different SSR among different plants. This is the first advantage. And second, there are many kinds of simple sequence repeat. Like we just said, 2 bases repeat, 4 bases repeat, and so on. So there are many markers that you can use. Third, even though distributed all over the genome, if markers have no diversity in the population, it is still not a good choice, because you cannot find differences among plants. With these 3 advantages, we chose SSR as our weapon to solve the 2 problems we've been dealing with: selection and hybridization. First, hybridization. We solve the hybridization problem, which is, in other words, to identify seed purity with high efficiency and low cost. How do we accomplish this task? First, we synthesized 300 pairs of primers that can amplify different SSR loci. You have to test a lot of primers until you find characteristic bands in paternal land and maternal land respectively. As to the example here, through screening of all the 300 primers, we intend to find primers that can detect differences between these 2 peppers. And finally, we found one. We found a pair of primers which can amplify the same SSR locus in both parental materials, but the lengths of SSR are different between paternal and maternal. Black lines here represent primers, and the other primer is here. As you can see, most part of SSR sequence is similar between paternal and maternal, with only one difference here, which calls the length difference between P and M. In the lab, so the electrophoresis would be like this. As you can see, the length of this SSR in paternal is longer than that in maternal. It's very easy to tell. And in the length of F1, as you can imagine, we found 2 bands representing heredity from both paternal materials. Thus, we can identify those true hybridization progenies in a batch of seedlings and calculate the seed purity very easily. Two tests were done before this method was brought into practical production. The first one is blind test. We asked our co-workers to put several maternal materials, which is represented by this symbol, maternal materials, into a batch of F1 plants. Then we extracted DNA and did the SSR detection. These are the results. The electrophoregram A here was tested with pepper BF in Chinese Baofeng, and asterisks represented maternal plants mixed designedly by our co-workers. We identified out them all. 3, 4, 5, 6, 7. And here, pound sign here was not maternal plant mixed by our co-workers. We speculated that it is a false F1 seed due to hybridization failure. Electrophoregram B was tested with pepper 7318. This is the name of the pepper. Again, we screened out all the plants mixed designedly. The second test was comparison between lab and field. We fetched 400 seeds out of one batch of F1 seeds. 200 seedlings were sent to the lab, and the other 200 were planted into field. With pepper BF, seed purity identified by visual inspection was 99.2%. And the seed purity identified by molecular marker method was 97.8%. Again, with pepper TJ2 in Chinese Tianjiao, mass and traditional method gave seed purity of 97.6% and 98.4% respectively. And what can you find from these two groups? Compare the numbers. Which is higher and which is lower? Yeah, sorry, lady first. Okay.
The higher ones are always coming from the field.
Okay. Okay. The gentleman? Yeah. Not only the two examples, all those later tests on other cultivars showed similar tendency that result with molecular marker show lower seed purity than the results of field inspection. In other words, if lab reports say that the seed purity is above 95%, you can sell it with safety because the results from field normally be higher than 95%. Now, passing the two tests, we feel confident to continue with large-scale screening of primers. After screening 9,800 times, we found in some 149 pairs of primers usable on top 29 most important pepper varieties in Anhui Jianghuai Seed Company, with an average of 5 pairs of primers for each variety. Question: if we want to calculate the seed purity, all we need is only 1 pair of primer, right? We only need 1 pair of primers to calculate 1 cultivar. But we screen 5 pairs of primers for each variety. What is the use of the other 4 pairs of primers? We'll talk about that later. As to pumpkin, we found 101 pairs of primers usable on top 17 most important pumpkin varieties in Jianghuai Company, with an average of 6 pairs of primers for each variety. This is very important. Okay?
I don't know if you're going to talk about it later, but how do you choose the primers among the around 100 primers? How do you choose the 6 pairs?
Yes, that's a very good question. At the time that we screened out 5 pairs for each variety, we think that exact question, we asked ourselves the same question. Which primer is the true primer that can be used to identify seed purity? At last, we decided to use the primer that gave us the result that is similar to the field result. Because you sell those F1 to farmers, they don't use math to identify seed purity, right? They use the traditional inspection way to identify the seed purity that you sell them. So we choose the primer that gave us the result similar to the field. If you use these 5 pairs of primers for the same variety, you will get 5 different results of the seed purity. I will talk about that later, and that is a very good question because it happened in reality. Thanks. I hope I have solved your problem.
Yes.
Okay, thanks. And the corresponding table of varieties and prime is shown here. This column is variety name, and these are primers, and these are annealing temperature used in PCR. And we are going to perform PCR experiment this afternoon by yourself. You will do the experiment. Within this table, it seems that we can solve all the problems on seed purity. We calculated our productivity. 2 technicians working for 8 hours can accomplish purity identification of 200 plants. I say this again. 2 technicians working for 8 hours can accomplish purity identification of 200 plants. Every batch of seeds needs to be checked for at least 200 plants. So our productivity was 1 batch per day. Then we asked our co-workers from the sales department how many batches needed to be checked every day to meet requirements of sales. They gave us a number. This is what we can do per day, and they gave us a number. This is what we need to do per day. We knew that there was a long way until we can solve the problem on seed purity. So we started establishment of a high throughput SPI, which stands for seed purity identification, high throughput. We analyzed the whole procedure of math to see where the time limits were. 6 hours were needed to extract DNA from seedlings. Then DNA was used in PCR as template. It takes nearly 2 hours to run a PCR program. It was clear where the time limits were. First, we tried to accelerate DNA extraction. A commonly used DNA extraction method was displayed here. Plant materials were subjected to manually grinding or grinding instrument like this one. It took at least half an hour to break cell wall in spite of using which method, manually or by instrument. After grinding, powders were subjected to extraction buffer like this one and a water bath of 60 degrees Celsius for 45 minutes to lyse cell membrane. Then organic reagent was used to extract DNA from cellular content. At last, high quality, very high quality DNA was obtained through centrifuge, washing and desiccation. As you can see, this method had cumbersome steps. How can we shorten time? It has so many steps. How can we shorten time? I have repeated for 3 times that the DNA is with very high quality. The DNA extracted by this method was of a high quality, complete, and pure. Complete means DNA fragments were very, very, very long, usually longer than 5 kilobases. But do we need DNA fragments to be so complete? If we want to do SSR detection, if you can recall that SSR is a kind of DNA sequence with certain DNA motifs ranging in length. Yes, very good. You remember? Ranging in length from 2 to 5 base pairs, repeated only for 5 to 50 times. So 5 base pairs repeated for at most 50 times is 250 BP, right? 5 times 50 is 250 base pair long, which is very short. And second, DNA purity. DNA purity was required if downstream experiments were gene clone, thousand blot, and so on. These experiments require very pure DNA. But we just need to check DNA results. We just need to check PCR results and no more else. We don't need to require such a high quality. So, considering these inspired us to abandon commonly used DNA extraction, which has so many steps, and considered abbreviated extraction, which use alkali boiling to extract, and pH adjusting to acquire DNA that is suitable for PCR reaction. Use alkali boiling to extract DNA, and second, adjust pH to make the DNA suitable for PCR reaction. Obviously, it's time and labor saving. If you want to know whether we succeed with this abbreviation method, we can maybe take a short break and I will show you whether we succeeded or not half an hour later, okay? So let's take a break and come back in half an hour. perform seed purity identification because you have to consume 200 seeds. But we have a lot of them, so 200 seeds is affordable for us. Then we tested purity identification with buds in quantity. As you can see, roots of germinated seeds of pepper TJ3 adapted alkali boiling method very well. We identified 3/4 F1 seed out of 94 seeds. So the seed purity of this batch was around 96.8%. Positive controls. Here, positive controls, parental and maternal. Paternal and maternal lines are also showed clear bands here. Germinated seeds of pumpkin N-008 gave us successful experiences too, with DNA template extracted from roots and primer CMT, which represent pumpkin. M224 is the name of the primer. With this primer, we succeeded in 102 PCR reactions. Again, 3/4 F1 seeds, this one, and this one were identified. Again, positive controls show the results here. Utilizing the abbreviated extraction method, which was composed of two main steps. First, distributing roots into centrifuge tubes and alkaline boiling. Second, transfer DNA from this tube into centrifuge into 96 well PCR plate for PCR reaction. We were able to shorten DNA extraction time from 6 hours to 1.5 hours per 200 seeds. After several times of practice, one of our technicians suggested that we can directly extract DNA in the 96 well PCR plate. So the final procedure went like this. After adding roots and 80 microliter of alkali, usually sodium hydroxide, into the well. Plates were put in warm baths for 99 degrees Celsius for 20 minutes. Then tris chloride was used to adjust the pH to 6.0. Because PCR reaction will fail if pH of DNA template is too high. Because you extract DNA with alkaline, right? So the pH is very high. You need to adjust pH to 6.0. Now, supernatant with DNA in it can be transferred to a new plate very conveniently with multi-channel gun, this one. This little adjustment shortened extraction time further to less than 1 hour per 200 seeds. Now PCR became the only time limit. We surveyed the whole process of PCR reaction. The total volume of a PCR is 20 microliter, composing of buffer, DNTP, and polymerase, which can be bought from reagents cooperation. Primers were designed by yourself and synthesized by company. We have just to say that we synthesized 300 primers, right? And DNA as templates was extracted from plants. Then PCR plate containing these was put into a PCR amplifier to go through this program. At a very high temperature, like 94 to 96 degrees, template DNA would denature into 2 single chains. Then at a lower temperature, around 55 degrees, primers bind to DNA, where temperatures rises to 72 degrees, polymerize here. Polymerize synthesize another chain of the fragment. After 35 circles, original SSR fragments will be amplified to a huge magnitude that can be detected through electrophoresis. This is a very time-consuming process, making the whole PCR program cost an hour and 45 minutes, which is too long for us. Our instrument supplier suggested that we could try a special PCR amplifier named Speeder. One of the main reasons that PCR programs take such a long time is temperature changing from 94 degrees to 55 degrees and then to 72 degrees. Speeder, this one, can save your time by accelerating temperature changing rate because the temperature control module of it was made by gold, metal with the highest thermal conductivity in the world, gold. And indeed, it could double PCR speed. However, the drawback was price. Roughly 4 times of ordinary PCR amplifier. Besides, it used a special kind of 986 well plate, which was also expensive than ordinary ones. To solve the problem, we have to figure out other ways. We surveyed the program again to find the reducible parameters. You can't reduce this one, the circle number, because you have to amplify SSR fragments to 2 to the power of 35 to visualize them. If the circle number was reduced to 30, for example, electrophoresis detection would fail because the number of the SSR fragments was very, very low. Thus, it seems that time in every circle is the only option. We tried reduction of warm bath time. The results are shown here. As you can see, canceling the first pre-denature step and leaving the rest, this one, the pre-denature step, it takes 3 minutes past, but now we cancel this pre-denature step. And leaving the rest, time parameters as original didn't fail us in PCR. The bands are still very clear. We made a series of reduction tests, realizing that time parameters were all reducible actually. Finally, we were surprised. We reduced, and reduced. Finally, we were so surprised to find out that 0 minute, 1 second, 1 second, 1 second, and 0 minute program would also give us a good electrophoresis, although not so good as the original one, but the bands are still good enough for us to get the results right. So now we have adjusted the standard program to this form to this form. Reduce the PCR time from 1 hour and 45 minutes to 24 minutes, which means we have raised the PCR speed as much as 4 times without buying expensive amplifier that's speeder. A side effect of speeding up was that bands became darker. To increase PCR quality, we screened PCR reagents. These are PCR reagents. We screened PCR reagents from several companies and finally choose one of them. It had the best performance under the adjusted program with reduced time. Then we conducted large quantity tests with both pepper and pumpkin. Upward, from the first one to the 100, is the electrophoregram from standard program. This is the standard PCR program. Lower is the one from adjusted program. With shorter warm baths, bands became darker as you can see, but it was still clear enough to identify single band, like this one, from double bands. Positive control work too. This is a test performed on pepper. And things were a little different as to pumpkin. When the program was adjusted to 0 minutes, 1 second, 1 second, and 5 seconds, 0 minutes, bands were too obscure to use back. We cannot see them. So we ascertained 0 minute, 2 seconds, 2 seconds, and 5 seconds, 0 minute to be the final setting. Under this adjusted program, differences between single band, like this one, and this one, between single band and double bands were distinctive enough. Also, again, positive controls work too. Now, we have solved the final time limit in the whole procedure. A high throughput seed purity identification method was established by reducing time of DNA extraction and PCR reaction. Brief summary. For the past decades, we've been using traditional method to calculate seed purity, which was time-consuming, and it requires experiences on phenotypes of varieties. It usually takes an amateur 3 years to be familiar with physical characteristics of all varieties. And phenotypes of plants can be easily affected by soil, sun, wind, and temperature, environment. Thus, the traditional inspection method is unstable. On the contrary, math can save a lot of time, especially when the high throughput identification system was established by us. We speeded up the whole procedure for 15 times. Second, the whole system is very easy to master. It only takes an amateur 2 days at most to learn all the operations. Third, DNA won't change by environment, so it is quite stable. And among all kinds of markers, we chose simple sequence repeat and synthesized 3 pairs of primers. Through screening, all of the varieties had at least 1 pair of usable primer, as I have showed you before. This variety primer table was just a foundation. Then we perform large-scale primer screening on varieties, on all the varieties that the company has. And with the newly established whole procedure, now we can cope with 15 batches per day. Before it was 1 batch per day. In China, 4 months from September to January are selling season, which are also the busiest season for molecular lab in Jiangwei Company. And last year, within 4 months from September to January, purity of 1,500 batches of seeds, every batch with 200 seeds. 1,500 batches of seeds were identified by only 4 technicians, which is unbelievable before. Now, we have solved the problem of hybridization, the seed purity. We then continued to focus on selection. Mass can be used to select false F1 seeds. It can also be used to select desirable plants. In the past, breeders crossed a susceptible cultivar like this with disease, but with high yield cultivar. With this one, a resistant but low yield cultivar. Progenies with resistance and high yield were tested and selected from a large population. This is a traditional selection. But with mass, molecular markers can be used like fingerprints to distinguish different cultivars. In an easy way, progeny with heredity from both parental materials can be screened out precisely. To gain this goal, you need 2 different cultivars, which are represented by their different phenotypes, and the markers closely linked to phenotypes. It is, however, not easy. The difficulty lies in the markers. You will find too many different markers between these 2 cultivars. If you can recall, we have screened average 5 usable primers for each variety. Did you remember? You find too many different markers between 2 cultivars, and it is very hard to tell which marker is related to your desirable phenotype. That 5 markers, which one is related to the desirable phenotype, like resistance, like high productivity, which marker is related to the phenotype you don't know. Because you selected 5 primers for the variety, but susceptibility and resistance is not the only difference between the 2 cultivars. If it is the only difference between the 2 cultivars, then those 5 primers that you screened out is related to the phenotype difference, right? But susceptibility and resistance is not the only difference between the 2 cultivars. Like leaf shape, fruit color, and plant height may all be the differences. And markers screened out from the 2 cultivars are linked to all those differences. It usually takes several generations to identify markers linked to your desirable phenotype. We started to wonder, is there a faster way to select good parental materials? Can it be possible to go from known markers to phenotype? Will this way be easier? In traditional way, it is from phenotype to markers, but we asked ourselves whether it is easier to go from markers to phenotype. So, speaking of markers, we have selected many molecular markers for many cultivars. So we asked ourselves, why don't we need so many primers, which is a question that gentlemen have raised last class, to identify seed purity when usable pair of primers would satisfy. What else can we do with so many primers? An experimental consideration came to us. What are the result differences of these primers? Will they give the same answer on the identity of cultivars? The answer is no, as you can see here. We found that there were different types of plant within one parental material if you use different primers to identify it. DNA of a maternal material pumpkin, ZMB in Chinese, 早蜜本, was used as template, and primer, MC3, and primer, M232, were used respectively in 2 batches of PCR. As you can see, majority were identified to be maternal, like these ones. They are identified to be maternal, while some had discrepancy between 2 primers. The results are different between these 2 primers. We named them type 1, type 2, and type 3, type 4. The same thing happened to maternal material pumpkin HJ2. There were 3 types of plants within the material. These are plants identified by 2 primers. So number 21, primer A says it is not a maternal, but primer B says it is a maternal. So the discrepancy between the 2 primers may help you to type these material plants. Again, same thing happened to pepper. 2 types and 3 types were identified from pepper BF and BC. Now we have markers between cultivars. How to get from markers to genotypes? We planted them. At seedling stage, we numbered the plants and amplified and applied multi-primer inspection on them. But we numbered them from 1 to 100. Then plants of the same type were planted together. We plant type 1 together and we plant type 2 together. Unified fertilizer and water management were conducted until fruit setting. During the whole period, characteristics of all aspects were carefully documented by us. At last, fruits were collected together and compared with each other. The final data proved that the marker to the phenotype way was a feasible one. The marker to phenotype way is a feasible one. I will give you 3 examples to show the efficiency of this thought. First one, winter squash, KBDB. We identified 4 types of plants within maternal materials and 3 types of plants within F1 generation, which is not shown here. The paternal materials seem to be quiet. This is the maternal. The paternal material seems to be quite pure. Then we plant them according to classification, type 1 to type 4, according to different types. Like this, the same field management was implemented to all of them. Growth status was documented meticulously until fruit setting. First, let's take a look at the appearance of the fruit. They all seem similar with each other, but let's take a look at the productivity. The productivity result was very interesting. This line is a paternal material. And these 4 lines are 4 types of maternal lines. These 3 lines are 3 types of F1 generation. Yes?
You're telling us look at some lines in the table, but perhaps you're trying to show them with the mouse in the computer, but nothing moves in the screen. Perhaps if you try to move the mouse with your finger,
Oh, the mouse didn't show?
No.
or you can use the pointer that you have. 中间那个 stir 上。
Oh, now you can see this red dot. I'll start from here. This line is paternal material. The first line is paternal material. These 4 lines are 4 types of maternal materials that we identified with 2 primers, type 1 to type 4. These 4 lines. And these 3 lines are 3 types of F1 generation. The genotype show illustrates PCR results here. P19M M121B means PCR results with primer. P19 was maternal. P19 primer says that these are maternal material. And result with M121 primer was double band. Then production per plant. How many this production per plant, and how many plants are there within this type. Total production, average weight of fruit, and the total number of fruits within this type are all documented carefully by us. Below are statistics of every single fruit. It's easy to notice that type 3 maternal material, this one, the red one, has the highest productivity among all maternal materials. Total production is the highest. And the type 3, F1, has the same quality. Total production is the highest. To explain this phenomenon, we checked for growth status differences among them. It turns out that type 3 maternal materials and type 3 F1 materials, seeds, all showed first female flower very early. We documented all the characteristics through their growth very carefully. So we have the data. First female flower. As you know that pumpkin first emerged male flower, and then the 10th to 14th flower may show the first female flower. So first female flower is a very important trait in squash production. Normally you have to wait until the 10th to 14th node to see the first female flower. If the first female flower shows up early, the fruit can mature early and set more fruits. You will sell your product earlier than other people. Thus, we finished the process from markers to a phenotype. First, we used markers P19 and M121 to categorize those materials. And then we find phenotype differences among them. So this is the whole process from markers to phenotype. And the phenotype happens to be a very important phenotype. The first female flower. It can solve the problem of late maturity. Harvesting early, you can sell them at higher price. This is meaningful to. This is the meaning to squash farmers. And to hybrid seeds manufacture, this is also a valuable trait. During the production of hybrid seeds, it is feasible to have female flowers waiting for male flowers because male flowers show up earlier than female, right? And it is feasible to have female flowers waiting for male flowers, while infeasible to have male flowers waiting for female flowers. So female flowers is more important than male flowers. So early female flower trait means productivity of hybrid seeds safely. Knowing this, our next step is markers-assisted selection for early female flower materials. We have now get ourselves a marker that is closely related to the early female flower trait. So we use it for selection. We believe that the probability of success is very high. And we are doing it right now. The selection of early female flower material. Another example is Chinese pumpkin, ZMB. Similarly, we identified 4 types with discrepancy between these primer MC2 and primer M232.
I think I'm a bit lost. When you talk about types, you're talking about cultivars, or type is referred to the marker.
The type is referred to the plants. because the results of the two primers, they show us 4 types, and we use that to categorize those plants.
Like 4 different combinations of plants in the...
Yes, exactly. And similarly, we plant them due to their different type. And finally, the phenotype differences among them was surface color. As you can see, these are greener and these are redder. Fruit coloring happened first with type 1. Coloring is one of the commodities in Chinese pumpkin. Similarly, with math, it's a goal to select coloring fruit. It still takes several generations to acquire stable materials, but with markers we believe that the process would be easier and with more accuracy. I have showed you 2 examples with pumpkin, and the third example is pepper. The interesting thing about pepper BF in Chinese is baofeng, is that we failed to identify different types within both parental materials. They are all pure with 2 primers, but we succeeded to identify 2 types of seedlings with F1 generation. This is very interesting. Although there were only 2 types, the phenotype difference was very significant. Can you find? Sorry, speak louder. Yes. Have you discovered?
I think, is that the first bifurcation? That's a seedling, or that's
The place where the first flower is placed.
the position of the...
No, it has nothing to do with this beautiful white flower. Take a look at this area and this area.
The tricoma.
Yes, you're right. Can you see those hair here? This has much hair and this has less hair, right? The exact name of this is trichome, and that lady has named it correctly. Trichome is the hair on the surface of a plant. Trichome is very important for plants. And type 1 was hairy, while type 2 had smooth surface. I will show you what trichome can do. Trichome, these are trichome, was a scientific issue studied substantially on many plants. Trichome, T-R-I-C-H-O-M-E, trichome, are studied substantially by scientists all over the world. These are scanning electron microscopic images. The reason that so many scientists are researching on it is that it is a weapon of plant to fight against insects. Yes, it's a weapon of the plant. Glabrous means glabrous means having no hair or similar growth. Comparing with hairy plants, glabrous plants are much more susceptible to insects, indicating that the hairy phenotype is a very valuable trait for vegetables. If you don't have any hairy, the insects may bite you and cause damage to you. With these examples, 2 from pumpkins and 1 from pepper, we conclude that with markers, we can classify one single parental material. It's different from seed purity. In the seed purity identification, we use markers to identify 2 materials, right? Paternal and maternal. But in selection, this step, we use marker to identify the same parental material. This means that our parental material is impure, actually, although it is used as a material, but it is impure. It is supposed that a material has to be pure to be used as a parental material. This is a very, very serious question. How many parental materials have this impurity phenomenon? This is a very serious question. If your material is impure, that means that the hybridization with it may be forced, may cause disaster. We surveyed and found that 2/3 of our parental materials are actually pure, with all the usable primers, while 1/3 of parental materials we use were actually not pure at all. Result of primer was 100%, but the others usually give low purity results. This is truly shocking, actually, because we used to believe that all our materials are very, very pure. Because we spend so much labor and time, did you remember this that I showed at the beginning? We spend so much manpower and time to select pure materials. And now, markers told us, you were wrong. They are not pure. They are impure at all. Yes, we were wrong. Those materials we thought to be pure turned out not to be pure. So the question came to us, why so many parental materials that are supposed to be very pure have such a low purity? We asked ourselves another question before answering this question. Why is there always a pair of primers whose result is 100%, with others showing very low purity? What does this 100% represent? Is it the phenotype that we've been selecting? Because we have now already know that a genotype is related to phenotype, right? So is this the phenotype that we've been selecting? So the purity is 100%. And the other are those phenotypes that we are not selecting. So their purity are very low. This thought gave us a possible explanation. When we were selecting materials, we focused on main commodities, like the shape, the color, and the taste of fruit, but we ignore those other traits, like hairy and glabrous. After checking all the plants produce uniform fluids. We thought that during this process, after that, we thought that the material was already pure. However, when checked with molecular markers linked with other traces exposed the truth that the materials are actually not pure in other aspects. Figuring this out, we continued our thinking on those pure materials. Those 2/3 pure parental materials. And we asked another question, are they really pure? We highly believe that the answer is no, although those primers that we have now say that the purity is 100%. But we highly believe that the answer is still no. This is a schematic diagram of pumpkin genome. We searched for SSR loci through the genome by utilizing a computer program. And finally, 88,177 SR loci were identified. And the primers we used in our so-called large-scale primer screening were just 300 primers. You remember that we synthesized 300 primers, which means the possibility of using much more SSR primers to identify those so-called pure materials. These considerations not only answered the question, are they really pure? We highly believe that they are not pure, but also answered this question: why so many parental materials that are supposed to be very pure have such a low purity? Actually, I hope you can understand that this is actually a fake question. It is not a question, because these parental materials were meant not to be pure. Actually, no parental materials was supposed to be very pure. Precisely speaking, parental materials from traditional breeding was supposed not to be pure. Final summary, okay? Final summary, by establishing a high-throughput purity identification system, we solved the problem of hybrid seed manufacturers, providing quality guarantee for sales. This is a problem with hybridization. And second, through multi-primer identification of parental materials, we realized that all of our materials were actually not pure. And we can use this to select. It is not pure, but it is not a disaster. We can use this to select new desirable traits, such as early female flower, such as coloring, such as the resistance to insects from existing materials. Okay, last section. This is the last take-home message. I know that after seeing all the contents that I have showed you this morning, the biggest concern of you would be, how do I use this technique with my vegetables, right? I was told that all of you are doing something related with vegetables. So maybe the most concern of you is, how do I use this technique with my vegetables? It is quite simple, actually. First, you start with this website. I have give you examples with pumpkins and peppers, right? But maybe you are dealing with other vegetables. So first start with this website. It is called Web of Science. And type in your vegetable here and add SSR to search for SSR markers in the genome of the vegetable. Then usually you will find research articles titled like this: Characterization of tomato. Here I searched tomato, which is a very important vegetable all over the world. Characterization of tomato SSR markers developed using back-end and cDNA sequences from genome databases. You don't have to understand all those information. You just need to ascertain the name of your vegetable, like tomato, and SSR marker, and genome. These three keywords exist in the title. Then you will get yourself a bunch of useful markers from the article. In addition, another good website that I want to recommend to you, if you are researching on these crops, these are all very, very important crops. Tomatoes, potato, pepper, Basmayana, which is the Latin name of tobacco, right? Tobacco, petunia, and eggplant. In a word, if you are working on solanaceae crops, there is a very good website that have already collected all the molecular markers for you. Find the submenu item of markers here. Now, under the menu button of search, then choose to show markers mapped in your interested plant. And within all kinds of markers, there's a list here. You choose SSR marker, which is the most recommended with comprehensive consideration of convenience, economic cost, and the repeatability of results. Leave all the others unchanged and hit the search button. All the available markers are already there for you to use. Click on the name of the marker. You will get all the information that you need to perform PCR. The aligning temperature, the sequence of forward primer and the reverse primer, and the size of the PCR product. Now all you need are two things. First, randomly select and synthesize 100 SSR markers. Second, equipment, just two apparatuses, PCR and electrophoresis, which are very easy to get. Next, screen those 100 SSR markers between two parental materials, 4 primers that show polymorphism between paternal and maternal plants. Now you can do at least two things with these primers. First, you can dramatically lower the cost of seed purity identification and ensure the safety of hybrid seed productivity. Second, screen markers with a large number of individuals within your material. Try to find differentiated types within your material. I'm sure you will find, as I have showed you today, that all the so-called pure materials are actually not pure. After classification of your material, plug them into field and observe the differences among different types. By doing this, you will make your materials more resistant to disease or show early female flowers. Thank you. These are my share on molecular markers in vegetable breeding. And if you have any questions that you want to discuss with me, you are welcome to email me with this email address. I'm looking forward to receive email from you. And that's all for this morning section. Thank you. And for this afternoon, we are going to perform PCR experiment, and I would like you to calculate the impurity. So please come back and shout again. 1:30 p.m. Okay?
A las 1:30 nos reunimos aquí a las afueras y esta barra nos llevamos al laboratorio, con el profesor.
I have a question.
Okay.
So, what would you think about doing